The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 68 of the eLife Podcast. I'm Chris Smith from The Naked Scientist. This month, why whale watching boats are making too much noise, what the muscles around your ears reveal about your real intentions, and the fish ingredient that combats heart failure. But first, Huntington's is a horrible inherited neurodegenerative disease. The symptoms normally begin to appear in a person's 30s, and they include abnormal movements and cognitive changes. They're caused by the progressive loss from the brain region known as the striatum of a population of neurons that respond to the nerve transmitter chemical dopamine. Blocking dopamine to reduce the activity in the cells can produce some symptomatic relief, but the side effects of doing this are very unpleasant. Recently, Queen Mary University of London's Peter McCormick has found that alongside the dopamine receptors on the affected cells, there are also histamine receptors, and blocking those secondarily quietens the response to dopamine and in experimental mice seems to halt the progression of the symptoms.
We discovered several years ago that the histamine receptor can modify dopamine receptors' ability to function. So we thought, could we take advantage of that characteristic and dampen the out-of-control dopamine by a histamine?
How does that work then? Is the histamine signalling to the same cells and it's just having an effect on the way those cells respond to the presence of dopamine?
What we think is happening is we've got a histamine radio tower right next to the dopamine one. And by targeting the histamine with a drug, we can dampen the output signal of that dopamine radio tower, which doesn't allow it to signal as well. So it's running interference, basically.
And why does this ultimately translate into a benefit for the disease process?
We think it slows down the ability of dopamine to signal, and those neurons don't fire as much, so they're more likely to stay alive and not die.
So the disease process is still grumbling along underneath. You haven't affected that, but what you have done is to control some of the ways in which it impacts on the vulnerability of the cells that would otherwise die.
That's correct. So we're not specifically targeting the Huntington's protein, and we're not fixing the disease, but we are able to slow progression. And the hope is, because there are quite a number of histamine compounds in the clinic, they might be repurposed for Huntington's.
How did you do this, though? Presumably this is not a clinical trial yet. This is preclinical animal data.
That's correct. So we have to initially show that our idea would work in cells. So we first worked in a cellular model of Huntington's, and then we later took that to a mouse model of Huntington's, and then we later showed from a brain bank, human brain bank, that the same target of these two receptors together existed early on in Huntington's patients.
So that suggests, were you to translate this to humans, because the same target's there as is in your experimental animals, it should work. But what sort of difference do you get in these animals with the rodent equivalent of Huntington's disease? What sort of a difference to the outcome and clinical course does your intervention make in these mice?
Yes, so what typically in this disease, especially in the preclinical model, you start to see neuronal death very early on. You start to see cognitive problems, so they have trouble remembering, short-term memory, long-term memory, spatial memory, and motor problems. All of those we were able to reverse by treating with a drug targeting the histamine receptor.
And when you say reverse, does that mean as long as the animals keep taking the drug, they just don't manifest any of the usual signs of the Huntington's disease?
That's correct. We saw no development of any of the symptoms of the disease, and we didn't see any neuronal death. And what was interesting is we did a very short treatment with these animals, and yet we could still see benefits a month after we removed treatment, suggesting that an early intervention with such an approach might really provide some exciting benefits.
The problem is that people who are prescribed some of these dopamine blocking drugs describe horrendous side effects.
Yes.
Is there a possibility, though, because the histamine receptors are there for a reason in these brain cells, Is there a
Yeah.
possibility that we're just trading one set of side effects for another, and your animals are still going to manifest cognitive problems or at least emotional problems, perhaps problems even staying awake because you block these receptors?
Now, we didn't go in depth in terms of the side effects, and that's certainly, I think, one of the questions that will have to come up. We didn't see also in the control animals any cognitive changes that were unusual. There certainly will be a risk, but I think that if you speak with a lot of patients suffering from this disease, it is quite debilitating. My prediction is these side effects would be much less than targeting dopamine receptor itself.
And is that the next step, to now formally evaluate this in a clinical trial with a cohort of Huntington's patients and put them on some of these antihistamines and see if you can change the progression of their syndrome?
First, what we would like to do is select the right molecule. I think the molecule we used in our preclinical experiments was very effective, but there are other candidates out there that I would like to test in a preclinical model to just really make sure that we've got the right one before we go into humans.
Let's hope he can make that work, particularly since it's soon going to be 30 years since the mutant gene formerly known as IT15 that causes Huntington's was first uncovered. That was Peter McCormick. Now, when I was little, my dad used to laugh at the young me trying to wrap my tongue around the line, "A noisy noise, a noise, a noisy oyster." Try that when you've had a few. But a new paper out this month argues that noisy noises also annoy humpback whales. Do the boats that take tourists out to watch whales, despite respectfully keeping a safe and sensible distance, nevertheless disturb the animals and potentially jeopardise their health and the long-term health of the industry itself?
So my name is Kate Sprogis and I'm a marine mammal researcher, and I'm an Australian, but I've been based in Denmark. When we did this study, we were trying to find out if the noise from vessels or boats impacted the behaviour of the whales.
Which sorts of whales?
We studied humpback whales because around the world they are one of the whale species that are most common. They're also the target for whale watching because they're so interesting to look at because they do a lot of breaching and pectoral slapping. And so we wanted to see if the noise from the whale watching boats impacted their behaviour or not.
When you say noise, is that people being rumbustious and sort of revelling on deck, or are we talking about the underwater noise made by moving bits of boat, propellers and so on?
We looked at the noise from the boat engines because the whales stay underwater for most of the time, and that's the noise that they're hearing. And how they perceive their environment is through sound, whereas ours is primarily through vision. So we use our eyes to interpret our environment, whereas whales and dolphins that live under the water primarily use sound.
And how did you actually monitor the change in the whale's behaviour and do it in a way that meant that by doing that monitoring you weren't changing the outcome of the experiment?
So how we set up our experiment was that we approached the whale with the vessel and we played back different noise levels. And to determine if the whale changed their behaviour or not, we actually flew an unmanned aerial vehicle or drone above the whale at all times at an altitude of about 30 metres, and we recorded video so then we could see if the whale had changed its behaviour or not.
And what sorts of changes in behaviour were you looking for? How did the whales respond?
Through the drone video, what we were recording are the number of breaths the whale would take, and we would also record if the whale began moving. They would normally rest on the surface, and then if there was a change in behaviour, they would increase the number of breaths that they took, but they would also start swimming away. And this was reflected in the drone video.
And putting all this together then, how does the different levels of sound map onto what the whale's response is?
When we drove past the whale and we played back a quiet noise, the whale would continue sleeping on the surface, whereas when we drove past the whale and we played back a loud noise from the vessel, from the boat, then the whale would actually change its behavior. It would start swimming away from the vessel. And if you put that into a whale watching scenario, what you actually want is the whale to stay there so that the tourists on board can see the whale in an undisturbed way and not in a way that you know that they're being disturbed from your presence.
How does this compare, the amount of noise that you produced, with the amount of noise that a whale-watching tourist boat would make? Was this a relevant level of sound intensity that you were exposing these whales to and then seeing them change behaviour in this way?
Yes, so the noise that we played back was a vessel noise, and it was within the range of other whale-watching vessel noise that we have also recorded the noise levels from.
In other words, then, the boats that are going out there to take tourists to go and look at these wonderful creatures are nevertheless causing them disturbance. Is that the interpretation, and we should therefore give them some guidance about how not to do that?
Yes, so right now on the whale watching guidelines globally, there are no noise emission standards. So a loud boat can be whale watching a whale at 100-metre distance, and then a quiet boat can also be at that same distance. However, the loud boat is going to have an impact on that whale, whereas the quiet boat won't. So if we can encourage that whale watching boats are quiet boats, then this is going to have less impact on the whales than a loud boat would.
And can you actually offer them some guidance as to what constitutes loud and quiet?
What we recommend is that vessels be quiet enough that their vessel is around the ambient noise level within their area. So vessels should be able to be recorded within the area so that they know what their source level or the noise level of their boat is.
You looked at humpback whales. Do you think this is true of other whales that people go and observe as well?
Yes, it can be used for other whale and dolphin species because they also use hearing as their primary sensory modality as they're underwater. So if there is a loud vessel, then they're going to be able to hear that loud vessel instead of seeing that loud vessel. So if they're a whale that is resting on the surface, then they're most likely going to be disturbed by a loud vessel compared to a quiet vessel.
Kate Sprogis. Now, sticking with sound, to our human ears, and a discovery that might make hearing aids work a lot better in future. We're all familiar with the way that our dogs and cats move their ears about to tune in to the source of a sound. They do it using muscles attached to the external ear tissue. Now, modern humans, of course, don't visibly do this, but we do still have scraps of those same muscles. And if you record the activity of these muscles, you can actually work out what we're trying to pay attention to, as Eva Higginbotham heard from Daniel Strauss.
Our faraway ancestors moved a lot the ears to orient their attention, and in the course of primate evolution, when we moved from a more nocturnal lifestyle to a more diurnal lifestyle, we became more eye-oriented. Evolution just decided that we don't need to swivel around our ears a lot. But still, there are these muscles, maybe they don't serve the original purpose, but they're still there. And so we measured the electrical activity of the muscles, and we used these signals to decode attention.
So you saw that electrical activity in the muscles around the ears could indicate where the person was paying attention.
Exactly. And we decoded two types of attention. One type is the exogenous attention. So this is what is driven, by the physical world outside, and the endogenous attention. This is the voluntary attention, when you say, oh, I want to pay attention to this, or I want to pay attention to that.
Did you have to do different experiments then for the exogenous and the endogenous, or could you do it all together?
Exactly. We did two types of experiments. One, the person was sitting there in a chair. There was a chin rest. They had to read text in front of them, and they were surrounded by loudspeakers. And while they were reading the text, suddenly there was a sound coming front left, front right, or from behind. And this was the exogenous experiment because we wanted to see how this new surprising sounds capture their attention. And in the endogenous experiment, we played two stories, either from the two front speakers or from the two speakers from behind. And the participants had to focus on one of the stories.
And what did you find in those experiments?
If we found out that the activity of the muscles that we just mentioned before really pretty nicely indicates the direction in which a person is paying attention to. And, it was really all the time there, this effect of the ears trying to move in a way to this particular direction. So there was this sustained activity over a longer period of time, basically for the entire listening period. And this was a very surprising result for us.
And does the electrical activity in the muscle mean that the ears were actually moving? Like, were the ears twitching, trying to get closer to the source of the sound? Is that what you saw?
Exactly. We had a stereo vision camera setup. Stereo vision means we had two high-definition cameras on both sides. So each ear was monitored by two cameras. And the stereo vision allows us really to see, the movement in the three-dimensional space. And indeed, we observed these movements in some subjects. We used also a technique called video magnification that allows you to see the movements a little bit better than the original video. In some subjects, we really saw pretty large movements. And they are really correlated and consistent to the muscle activity. So you really see the nice correlation between the muscles being active and the ear moving this direction.
That's really amazing. So now we know that our ears are moving in response to where we're paying attention. What can we do with that information?
There is an application if you talk about hearing aids. Modern hearing aids, they have these directional microphones, so they should amplify the sounds in a particular direction. And if you're in a conversation, you look at somebody, but then you listen something here, maybe you focus your attention over there. And this can be realized by this technology. So these directional microphones really follow the listening intentions of the user. So that's a very friendly human-machine concept. You really have your intentions and the machine does what you want. It's really, I think, a big promise for our findings here.
Sounds good to me, and what a terrific result. Eva Higginbotham was talking to Daniel Strauss. He's at Saarland University. This is the eLife podcast. I'm Chris Smith. Still to come, how to get that elusive professor's post. First though, oily fish is claimed to cut the risk of cardiovascular disease. Indeed, populations with a high fish diet do have lower heart disease risk. Why though was something of a mystery, but now researchers think that one molecule, called trimethylamine oxide or TMAO, might be the key. It has a checkered history though, because previous work had labelled this something of a metabolic bad boy. It turns out though that was incorrect. The finger had been pointed at the wrong molecule, and Marcin Ufnal has instead discovered that TMAO is powerfully protective against heart failure, an effect it achieves by acting as a diuretic to reduce the workload on the heart and prevent fibrous tissue from stiffening the muscle.
So our interest in trimethylamine oxide started a few years ago when the paper was published in New England Journal of Medicine that people who suffer from cardiovascular diseases, they have high level of trimethylamine oxide. And we were wondering, how is it possible that seafood has a lot of trimethylamine oxide? And at the same time, if you eat seafood, it's good for your health. So in the next series of experiments, we found out that trimethylamine oxide actually is beneficial, whereas what is harmful is its precursor, trimethylamine. And trimethylamine is changed into trimethylamine oxide by our liver.
What is the trimethylamine oxide doing in the seafood then?
Trimethylamine oxide is called osmolyte. So it protects cells of animals from high osmotic pressure and deep sea animals from hydrostatic pressure in deep waters.
Is there potentially then a role for it in human health? Because there are certain tissues in our body which do, from time to time or periodically, moment to moment, experience extremely high pressures. And relevant to the cardiovascular system, I'm thinking every time my heart beats, especially if I'm running around, it's going to develop extremely high pressure in the muscle cells. So could this have a role in protecting the muscle cells then from those high pressures?
Well, actually, this was our hypothesis. But our experiments show that actually it's not the correct pathway. And the beneficial effect of trimethylamine oxide seemed to be associated with osmotic pressure. And in our case, this was like a rat model of heart failure. They accumulate too much water and too much salt. So giving trimethylamine oxide, we increased excretion with urine of salt and water. So actually, trimethylamine oxide worked as diuretic.
Talk us through the experiments you actually did then to prove that that was the case in your rats.
We performed 2 experiments in rats with hypertension and in 2 models of heart failure. And in both experiments, we treated rats with trimethylamine oxide for 1 year. The rats were given this trimethylamine oxide with drinking water. We tried to give them a dose that would be similar to the dose of trimethylamine oxide that would be consumed if you were just on seafood all the time.
And then what, you're following up the rats to see if there is an obvious morbidity and mortality outcome difference in the animals that eat the normal rat chow versus the animals supplemented with what would be rat seafood.
Yeah, and it was really striking for us. Rats that were receiving trimethylamine oxide all survived, whereas in a control group, only 66% survived. Also, other parameters such as blood pressure was lower in animals on trimethylamine oxide.
How did you link the outcome, this dramatic reduction in mortality? That's a third down. That's very considerable. How did you link that to it being the trimethylamine oxide, the TMAO, and what mechanism do you ascribe to it to cause that dramatic reduction in mortality?
In both of these experiments, we observed increased urine output, and we were wondering whether this might be related with the final outcome. So we designed another experiment in which we looked at the effect of trimethylamine oxide on urination. And we found that rats that were treated with trimethylamine oxide acutely with higher doses, they had significantly increased urine output, significantly increased excretion of water and excretion of sodium, which was very similar to that what we observe after administering drugs that are called diuretics.
What did this do to the heart itself? Because we know that if you look at heart tissue in people with heart failure, there are various changes that happen over a period of time, which, okay, it's alleviated by taking drugs like diuretics to reduce the amount of load on the heart, but there are other changes that happen. The heart remodels itself to compensate, doesn't it? So do your rats also respond in the same way to the TMAO when you give it?
Yeah, these rats had lower fibrosis. So in general, when you have failing heart, the cardiomyocytes, the muscle tissue, is substituted by other tissue, by fibroblasts. And in our rats that were treated with trimethylamine oxide, we had lower fibrosis, and we also had better parameters, hemodynamic parameters of the heart.
Do you think that this could translate to humans then? Obviously, it suggests that the reason people who have a very seafood-rich diet, and I'm thinking people in Japan, people who are like Inuit, who have a correspondingly low level of heart disease, might be our natural experiment that's already happening. But do you think that this could be a therapy for humans?
I hope it could be a supplement for a diet that does not have it. We could start with doses that will be just similar to doses that are taken by people who eat a lot of seafood.
Well, I shall order an extra portion of sushi for my lunch. Marcin Ufnau there, he's at the Medical University of Warsaw. So, you've got your PhD, you've done a postdoc or two, and you have pumped out papers and pimped your CV so you have a resume to die for. Now, you want that tenured post in your dream institution. So how do you get it? And are you making yourself as marketable as possible? Amanda Hagee and her colleagues wondered exactly the same thing as they went through all this, but they were frustrated by the apparent lack of transparency and mentorship that they encountered in science, so they decided to set up a survey to find out whether they were the only ones and what really does need to change.
We're going through and there's all these communities and Twitter and all these advice pieces out there of how you get that job, but there's no numbers. And so we wanted to make the numbers. So we made a survey from the people that are actually trying to be a professor. We got them to put their numbers into the survey and then looked at the correlations and comparisons between different people.
And what does the survey show? What did it reveal?
So the survey shows that essentially there's no super clear path to becoming a professor. Those numbers, all the little metrics from training, help to a certain degree, but nothing is like a super good predictor of what it takes to be a professor. So there's no one clear path. There's a lot of different factors. One of the biggest ones that we saw was the number of applications you put in is a strong correlate for how many interviews you get, which is then how many job offers you get. Applying to more places helps.
How many people actually filled in the survey? And do you think that what you found is representative of the pool of people, i.e. it includes people who are successful, but also people who are not successful? Because I put it to you that if you end up with everyone who's disgruntled filling it in, you're going to get a distorted picture. Or if you get everyone who's Einstein filling it in, you're going to get a distorted picture.
Right. We got over 300 responses, and it does have a bit of a survivorship bias to it in that our pool of those 300 people, about 60% of them ended up getting a job offer to be a professor. And that's pretty high. And that makes sense because it was born out of this peer mentoring group where everybody kind of knew they wanted to be a professor. So all those people, they're already looking at this career, they're already kind of driving down this path. But I will say, even though we do have this bias, there was not a single positive comment about the process itself, even though these are the people that are getting jobs.
And did any themes keep on emerging as repeatedly being said as this is a downside, this is a downside, this is a problem? I.e. things that we could tractably say, well, there's our intervention point.
Yeah, we've had this conversation a lot. One of the things that was repeatedly reported was this lack of mentorship and a lack of feedback. There's, trainees, they're working underneath someone that's supposed to help them become this professor. The people that are making the hiring decisions aren't really telling people what they need to become that professor.
Is the survey not just a bit one-sided in the sense that, okay, you've got the opinion of people who have or haven't made it, the people who are on the rough end of the interview process? Wouldn't a more valid approach be to go to the people who sit on these panels and ask them for examples where they've definitely got it right and definitely got it wrong? Because then you can see whether or not they really are asking the right sorts of questions. Because we've all hired people that we regret. I mean, it happens.
Yeah, and I think that's a really interesting point. It's something we tried to get at a little bit. We did do a survey of hiring committees. And it's true, like the things that the applicants think is important and those metrics and those numbers of what the applicants have didn't exactly match up with what the search committees say they value. And so that disconnect is a really interesting and a really important spot for improvement.
It's something, though, of a buyer's market, isn't it? And the market knows it.
Yep. It's definitely trending in that direction. One of the things we cite early in our introduction is that we're continually improving more and more people with PhDs. And all these people are coming out of this training. But the number of faculty positions has not really increased, so it's just kind of driving this hyper-competitive environment. We can just keep pushing the envelope of what we can ask. And we're seeing that with papers that are published. That's been well documented, that the number of papers people are publishing at the point where they're applying to be a professor is increased astronomically over the last 20 years.
Because on the one hand, science is the beneficiary. If you've got loads and loads of good people applying for a job, you can have the pick of the bunch and you're going to get good people. So that's wonderful. But then on the other hand, my concern is if the system is driving people to think in the sort of way that the system is making people think, i.e. I've got to have loads of publications, then people aren't necessarily thinking like a scientist. They're thinking strategically like a career person. And that isn't necessarily the same thing. And I think back to, I read John Sulston, who got the Nobel Prize for sequencing the human genome and the wonderful work he did with C. elegans. I read his book. At one very poignant point, he says he'd been in post for about 10 years before someone said, well, really, we ought to write a paper about some of this work, John. You wouldn't see that happening today. And this is someone who went on to get a Nobel Prize.
Yeah, it's interesting how much it's changed, the landscape with this like hyper competitiveness. And I don't disagree that we're maybe doing a disservice to science by making this super hyper competitive, hyper stressful environment. Because the other aspect that you see a lot of people commented when we talked about their feelings about the process is anxiety. And I struggled with it when I was applying too, is that we are taking these people that are super, super smart and just exhausting them and making them super stressed out just to get over this hump of trying to continue to have a career in science.
Well, I'm glad you brought that up, because my next question to you was going to be, when you're in the position of being on these panels now, you're going to be in a position to change things. So do you not think that because a lot of people have now come through this, been stressed to hell, okay, they've paid a high price, but now they're in a position to fix it, and they will?
Yeah, I really hope so. I plan to. Hopefully, we get this generation of scientists, which I'm hoping is my generation. We all care about the culture change, and then we all bring it forward with us as we go.
Well, good luck, Amanda. That is Amanda Hagee, who is now in her, hopefully, dream job at the University of North Dakota. And that's it for this month. You've been listening to the eLife podcast that's produced by The Naked Scientist. There are previous editions, references, and also full text transcripts for these programmes, as well as details on how you can subscribe to this podcast at nakedscientist.com/eLife. And incidentally, The Naked Scientists also publish a weekly science programme that covers the latest leading science stories. And in recent programmes, we've explored the long-term symptoms of COVID-19, and marking the start of September, we've just published a detailed investigation of where the novel coronavirus came from. You can find that at nakedscientist.com/podcast. I shall be back next month with another look inside eLife. Until then, though, from me, Chris Smith, thanks for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.